Welcome to a new episode of Drops of Science, the multilingual podcast that brings you the latest news and discoveries from the world of natural sciences. Before we begin, we'd like to share with you a small change in the podcast format. From now on, the news you will hear are going to be the same ones collected each month on our blog. This way, you can choose whether to read them or listen to them, depending on your preference. But the podcast won't be just an audio version of the blog. Each episode will always include one extra story, designed exclusively for our listeners and not published on the blog. And now, let's dive into the news we've selected for you this month. Science has repeatedly confirmed that the average temperature of oceans, seas and lakes is rising as a consequence of human-driven climate change. What is less clear is how temperature extremes, both low and high, are changing. In other words, yes, waters are warming, but how are the peaks of heat and cold shifting? A group of researchers from Michigan tackled this question by studying the North American Great Lakes, a system that contains about 20% of the planet's surface fresh water and that, since the 90s, has experienced dramatic increases in temperature, with consequences for ecosystems, fisheries, and water quality. To analyze them, scientists used a three-dimensional model similar to those employed for oceans, adapted to the complex geography of the lake system. They then applied the trending, the removal of the long-term warming trend, for the period going from 1940 to 2022, to isolate internal variability. This way, only relative oscillations remain, unaffected by progressive warming. Results showed that the Great Lakes are not only warming but also becoming more variable. Compared to the 40s, the range of variability has grown by 25–40%, depending on the lake. Both cold and hot extremes have increased, with particularly sharp jumps since the 90s, considered true break years in climate dynamics. Extreme events have also risen. Heatwaves increased by 85–158%, while cold peaks by 168%. In some winter and spring months, Lake Superior recorded spikes above 300%. This means that today the lakes alternate between exceptionally hot and cold periods more frequently. A key driver of this variability is the drastic reduction of ice cover, which has shrunk by over 70% in the last 40 years. Ice acts as a lid that stabilizes water. Without it, lakes absorb more solar energy and become more vulnerable to climate fluctuations. Added to this is the influence of major global atmospheric cycles, such as El Niño or the Arctic Oscillation, which explain turning point observed in historical series. Unfortunately, lakes with less water and depth than the oceans cannot buffer these variations and respond more sharply to atmospheric changes. Scientists hope that by understanding these dynamics, we will better predict future scenarios and design effective management strategies, preserving the natural cycles of ecosystems. Let's stay in the aquatic environment, but move into the depths of the seas, where for over a decade an emergency has been unfolding, leading to the drastic decline of many starfish species. The culprit is the sea star wasting disease, which initially manifests with loss of appetite and lethargy by the animal, followed by the appearance of rapidly spreading white lesions. Surrounding tissues deteriorate, the water vascular system fails, and the sea star loses its grip on the substrate. The arms twist, tissue stretches appear, they detach, and the body collapses into an unrecognizable soft mass, almost a sea star mush. The disease is highly contagious. One of the most affected species, the sunflower star, Pycnopodia eliantoides, has lost 91% of its population, earning a place among the critically endangered species on the IUCN Red List. This decline has triggered a chain reaction in marine ecosystems. These stars are major predators of sea urchins. Without them, urchin populations have exploded, devastating kelp forests, which are essential for absorbing carbon dioxide and maintaining marine ecosystem health. For years, researchers have been trying to identify the culprit behind this serious crisis, and finally, an answer seems to have arrived. A large team of North American scientists raised healthy specimens of Pycnopodia, kept them in quarantine to ensure there were no ongoing infections, and exposed them to various potentially contagious conditions: immersion in water from tanks with sick individuals, injections of tissues or coelomic fluid, and direct contact. The disease only spread with unfiltered or unheated material, suggesting the culprit was a living microorganism larger than a virus. The next step was sequencing the microbial communities of 16 healthy and sick sea stars. Among more than 55,000 identified bacteria, one stood out: Vibrio pectinicida, abundant in sick stars and almost absent in healthy ones. To confirm, researchers isolated the bacterium and infected healthy sea stars. The disease reproduced in all its stages, providing definitive proof. This discovery opens the possibility for breeding programs to increase tolerance to this bacterium and targeted antibiotic treatments. Identifying it is a decisive step toward the recovery of these species, bringing hope back to sea stars and ecosystems that depend on them. For the next news, we won't move much in space, since we're still in the water, but rather in time. If you were asked, what is the oldest scientifically recognized arachnid, your answer would probably be Parioscorpio venator, which lived during the early Silurian period, between 437.5 and 436.5 million years ago. The previous record holder was the Scottish Dolichophonus loudonensis, which at 434 million years old is 1 to 3 million years younger than Venator. Until a few days ago, that would have been a perfectly acceptable answer. However, a recent discovery has pushed the origin of arachnids, encompassing not only spiders but also scorpions and ticks, even farther back in time and, surprisingly, placed it in the ocean. The turning point came from the study of an ancient fossil, Mollisonia symmetrica. Initially considered a primitive marine chelicerate, the Mollisonia fossil, dated approximately 500 million years ago and preserved at the Harvard Museum of Comparative Zoology, was examined using advanced optical microscopy. Its segmented body, similar to that of a scorpion, with a rounded carapace and 6 pairs of appendages, already suggested a relationship with modern arachnids. But the real secret lays in its nervous system. Through the analysis of neuropils, regions of nervous tissue between neurons, researchers identified a remarkable similarity to those of today's spiders and scorpions. Mollisonia symmetrica, which lived in an aquatic environment, had the prosoma, the anterior part of the body, with segmental ganglia arranged in a radial pattern, and an unsegmented brain sending nerves to appendages resembling chelicerae. Most astonishingly, it exhibited an anteroposterior inversion of the brain compared to insects, crustaceans, and horseshoe crabs, as if it were flipped, just like in modern spiders. This unique arrangement, found only in arachnids, provides modern species with superior control over their many appendages, which is fundamental for the agility, speed, and predatory skills that still define them. The discovery therefore suggests that the earliest arachnids evolved in marine environment, not exclusively on land as previously believed. Traditionally, it was thought that the first creatures to conquer land were myriapod-like arthropods and some primitive insects. Now, thanks to this discovery, researchers suggest that an ancestor of arachnids, similar to Mollisonia, may also have made the transition to terrestrial life, preying upon other ancient creatures that already inhabited the emerging continents. Now we move to Mexico, considered one of the biodiversity hotspots for pine species. More than 60 taxa are found there, concentrated mainly in temperate mountain areas. This richness has led a group of researchers from the state of Durango to ask how pines will respond to the constant pressure of climate change. Their focus was on Sierra Madre Occidental, the country's largest pine forest reserve and one of the most threatened ecoregions in North America, due to both deforestation and climate pressure. The Sierra Nevada Occidental is home to more than 20 pine species, and future distribution models were developed for 19 of them. These species distribution models combine occurrence records with environmental variables to project where species could thrive in the future. A total of 32 environmental variables were used, and the SSP245 scenario was applied. This represents a pathway of moderate climate change, with growing economic development but limited climate action, predicting a temperature increase of 2.9 Celsius degrees over the next century. The analysis not only forecasted the fate of each species, but also identified areas of high species richness, helpful for planning new protected zones. Only 3 species, Pinus chambroides, Pinus chihuahuana, and Pinus engelmanni, are expected to maintain a broad potential distribution, while Pinus brachyptera and Pinus iocorensis could suffer the most severe contractions. 18 of the 19 species are projected to lose range. The most alarming case is Pinus brachyptera, which may lose 97% of its suitable habitat by 2040 and disappear by 2060. Others may decline by 2080 and slightly recover by 2100, or shift to higher altitudes. However, the rapid pace of warming raises concerns about their ability to adapt. These results are worrying, especially considering that only a small portion of the most biodiverse areas are currently under protection. Timely conservation efforts involving local communities are crucial to safeguard this irreplaceable ecological and economic heritage. We wrap up today's news by talking about the glasswing butterflies, whose common name comes from their transparent wings decorated with distinctive color patterns. This combination makes them both hard to spot in their environment and recognizable to predators. In fact, the species living in the same area look incredibly similar to one another, so much that they discourage birds from eating them. In fact, their colors signal their toxicity. An international team of researchers set out to study the rapid radiation of 2 butterfly genera, Mechanitis and Melinea, to understand what drove it. In evolutionary biology, the rapid radiation refers to the phenomenon in which a lineage diversifies into many different species over a very short period of time. These 2 genera are among the most complex to classify, since their species often look morphologically identical, not just in coloration or external morphology, but even in genital traits. Despite this similarity, they have diversified into numerous species. Another particularly striking feature is that they do not all share the same chromosome number. While most butterflies have 31 chromosomes, glasswing butterflies range from 13 to 30. To clarify this, the researchers sequenced the complete genomes of 135 Mechanitis and 109 Melinea individuals from South and Central America. This allowed them to confirm species status for most known taxa and to elevate some subspecies to full species rank, resolving several cases of ambiguous classification. They also found that evolutionary trees built from mitochondrial DNA, inherited only from the mother, did not always match those based on the whole genome. This mismatch can be explained by ancient genetic mixing, what biologists call introgression, the transfer of DNA fragments from one species to another through hybridization. The analysis further revealed that the diversification occurred very recently, within about 1.3 and 1.4 million years, with one of the fastest speciation rates ever recorded in neotropical butterflies. The Andes Mountains played a key role here, acting as a geographic barrier that separated sister populations on opposite slopes or at different altitudes, limiting gene flow. On top of that, numerous chromosomal rearrangements reduce the viability of hybrids, reinforcing reproductive isolation. The extraordinary diversity of glasswing butterflies is therefore the result of multiple factors that together consolidated the birth of new species. And although to us they may all look the same, they have a very precise way of recognizing each other. Each species produces unique pheromones that only their own kind can detect. So, while we might struggle to tell them apart, among themselves there's no room for confusion. This concludes today's episode of Drops of Science. Thanks for staying with us. See you soon, and good science!